Hello, in this video we will discuss about the blastulation and gastrulation to the neurulation. So collectively we will discuss in this manner. So in this way, after the fertilization, the blastula formation, and which that is the previous video I had explained about that. So let's begin to understand from the blastula. So what is the blastula? Blastula mean here is I am drawing the graphic for understanding. This is the upper chamber is known as amniotic sac, while the lower chamber is the yolk sac, which that is the precursor of umbilical cord, and on the other hand the endoderm, while the amniotic sac is required the ectoderm. So this is the epiblast and hypoblast is the disc-like structure are present between both chamber, and it is known as the bilaminar disc. So this is the yolk sac, which that is create the endoderm because it is endoderm, and also the umbilical cord production, umbilical cord creation. So in this way, the it is the allantois precursor, while this is the we are understanding the bilaminar disc between the two chamber. This is a bilaminar disc contain epiblast and hypoblast, which that is covered the downward, the hypoblast covered with the yolk sac, while the upper is the amniotic sac, epiblast. So in this way, this is a primitive node and primitive streak will be formed. In this way, remember, this the ectoderm. Basically, it is the ectoderm because the amniotic sac covered with the ectoderm. So it is the precursor of ectoderm will invaginate, and this is the invagination process. And in this way, the invagination will the ectoderm will move inside between the ectoderm, epiblast, and hypoblast. So this is the pink color showing that is this is the mesodermal area, which that is the differentiation of the cell and modification of the cell from the epiblast will move inside is known as invagination. So this is the primitive streak formation, and this is the mesodermal area, and this is the tail and head. Head is the primitive node containing head cranial area, while the tail containing a caudal area, the caudal side. So let's begin to understand here from the process via a cross section. So this is the cross section. Basically, is the it is the sagittal cross section. This is a sagittal area, which that is basically is the primitive streak is showing in the figure, which that is contain a primitive node. So in this way, the invagination after the third week. So this is the third week. That is the lateral. Basically, is you looking like this and contains yolk sac, and this is the ectoderm, which that is composed with the amniotic sac is the precursor of amniotic fluid. So this is the primitive streak formation, and this is the blastocele inside. We can say this is the blastocele is the precursor of amniotic fluid, amniotic sac. So this is the I am drawing the line which that is showing the head is the cranial area, right side, and the left side is the caudal area. So this is the endoderm formation which that contains yolk sac. Because it is the because it is the yolk sac which that is covered with the endoderm. So let's begin to understand further processes. So here is the head side. This is the lateral cross section which that is showing the head side, which that is invagination. It is the process is known as invagination, and this is the cross section and show the endoderm, and this is a mesodermal area, which that is the modification and differentiation of the cell will move inside, and the differentiation of the hypoblast will be occur, and in this way the mesoderm formation. This mesoderm modification will lead to also produce the notochord after the clearing, after the closing of the primitive streak. When primitive streak closed, but the primitive node are here, but the notochord which that will be formed due to the primitive streak closing. So after the closing the primitive streak, the notochord formation, which that is basically is the precursor of spinal cord. So here is a median mesoderm formation and lateral mesoderm formation due to the invagination of the cell of the epiblast. So that this is the lateral mesoderm which that will form the peripheral muscle and bones, while the median mesoderm which that is required for the production of other things, the heart as well as lung. So this is the endoderm and here the yolk sac. So you can see this is the ectoderm will cover here. After the next step, this will be covered. This ectoderm will be covered whole body because it is the skin production and the hairs as well as the outer covering. So that is why the ectoderm will be covered. Here you can see the blue color. So the neural groove will begin to form and neural fold will be occur in the epidermis or ectoderm. So this is the neural plate is used for the peripheral nervous system and central nervous system formation, the precursor of peripheral and central nervous system when neural plate formation. And neural fold will be occur which that will create the central nervous system. So the endoderm is here and this folding will lead to yolk sac will be different separate, and the notochord is here and the median and later mesoderm is present, peripheral mesoderm. So in this way you can see here is basically this process. This process is known as the neurulation formation, while that is the previous is the formation of three ectoderm, mesoderm and endoderm formation is known as the gastrulation. Gastrulation means the three germinal layers form. So this is the caudal and here is the head is the brain side, while the caudal side, this is the cranial is the brain side head and the caudal is the spinal, this is the tail side. This is arise the spinal cord, while the cranial area which that is basically arise the brain. So let's begin to understand the further processes of the neurulation. So this is the going to neurulation. We are understanding the neurulation. So the neural fold will be formed, neural plate will be formed, which that is basically used to enfolding, and after this enfolding, this process is the precursor of neural tube. So the neural tube formation will be occur. So this is the next process. In this way here is the median mesoderm and peripheral mesoderm will be formed, while this is the neural tube formation will begin to complete it, but here is incomplete, and this dark blue color showing that neural crust formation. We will also understand about the neural crust formation. This is basically ectoderm, so remember, and this is endoderm in the yolk sac cover, and the neural crust which that is used for the peripheral nervous system, the ganglion, the dorsal root of ganglion of the spinal cord and ventral root of ganglion, and also the sympathetic nervous system and peripheral parasympathetic nervous system formation, as well as the adrenal medulla formation. So this is the mesoderm and endoderm, and this is the future umbilical cord will be occur when yolk sac will be separate. So this is the neurulation process. In this way, the neural groove formation, and this neural groove is basically cover and complete. The folding will complete and bind, and this is the anterior neuropore and the posterior neuropore formation, while all covering, and in this way the somoid formation. So remember this mesoderm is known as the somoids, the lateral, while the inside is basically the endomesoderm. So this is the lining which that is that segmented lining. It shows that somoid, which that is the precursor of the bones formation, for example spinal cord as well as vertebral column, and this is the brain side, and this is the mature neurula. This is the neurula, mature neurula, and in this way here is the brain. So between the somoids, the spinal cord is present. The somoid is basically is the mesoderm, which that is the, and here is a notochord formation. The red color is showing in the figure. While on the other hand here you can see this is the neural crust formation, and this is the invagination of the ectoderm. So in this way, this is I am drawing here is you can understand this is the cross section, and will separate the neural tube and as well as neural crust. Neural crust is used for the peripheral nervous system, while the neural tube is used for the central nervous system. And the notochord is basically used for the vertebral column because it is arise from the mesoderm. And the yellow color is showing that the median mesoderm, which that is arise and to produce heart, lungs, as well as other liver. So on the other hand, here is a neural tube, which that neural tube is basically is the precursor of central nervous system, and this is the mesoderm, the peripheral. White color showing with the covering of the red line is the mesoderm peripheral, while the median mesoderm is a yellow color showing the figure. And this is the endoderm, which that is basically covered by the gut, and this is the gastrointestinal tract will be developed around the endoderm. So this is the endoderm formation, and the separation of the yolk sac, which that will become the future allantois. This is the umbilical cord, and this is the neurula. So remember, after this here is neurula. See, this folding will be occur. This folding is the median folding as well as the lateral folding. Basically is, you can see, this is the folding, and this is the basically folding of the embryo, and this is the amniot, future amnion, and amniotic fluid field, which that is used for the shock absorption of the fetus, and this is become a the embryo will become a future fetus. And this is the endoderm and the mesoderm, and the basically here is the endoderm, which that is contained digestive tract, and this is the allantois. This is the future umbilical cord, and this is the uterine wall, which that contain a blood circulatory system for exchange of oxygen and carbon dioxide between the fetus and the mother. So this is the amniotic fluid. So remember, this is the median and later folding, which that will be occur. Basically the tail and head will be also fold, as well as the median folding mean the basically is the also embryo will fold together. So thanks for watching. Please make sure to subscribe.